So I'm going to tell you about this publication that just came out about 2 weeks ago in Cancer Cell, in which we found a novel, I think novel, function of the PTEN tumor suppressor gene in the nucleus, which we believe we can leverage for therapeutic intervention in glioma patients. And as the title says, it says leveraging PTEN nuclear function for therapeutic intervention in glioma. I was thinking about maybe an alternative title for this. And so I came up with this novel function of PTEN in the nucleus. What's a nice lipid phosphatase doing in a place like this? And the reason I wanted to show this title is I've spent a good part of my research career in San Diego investigating PTEN from the perspective of its canonical function, which is as a lipid phosphatase in the cytosol. And so PTEN controls activation of the PI3 kinase pathway by blocking the production of a very important second messenger known as PIP3. And I'll tell you a little bit more about that. But what we now think is happening here is that in addition to the cytosol function, there are some unique functions of PTEN that don't require its enzymatic activity, and these happen in the nucleus. Okay, so in my lab, we function on, we focus on glioblastoma, among other types of brain tumors. And as illustrated here, GBM is the most common and aggressive of all primary brain tumors. And it basically accounts for about 50% of all primary brain tumors that occur in adults. And it occurs or it arises from glial cells, whether those might be neural stem cell in nature or mature astrocytes. I think the jury's still out on that. Oncogenic systems can show that either sources of cells can serve as a cell of origin for these types of tumors. Now, in the US, there's about 15,000 cases of GBM per year. Really, it doesn't sound like a lot, but the fact remains that about 80% of these individuals will die within 14 months of diagnosis. So the mortality rate for GBM is extremely, extremely high. Risk factors for GBM, we really don't know too much, other than Caucasians and Asians slightly higher than other populations, men slightly higher than females. And that's about it. That's really what we know. And so why is this tumor so difficult to treat? And I think there are really four main reasons. It cannot be surgically cured. These tumors are incredibly invasive. So the best neurosurgeons, such as Clark, really can't take out the entire mass of the tumor or all the areas of the tumor in which it has invaded. Secondly, there's no early detection for this type of cancer. Unlike other cancers such as breast and colorectal cancer, there's really no test to say that this individual might have a tumor developing. Thirdly, these tumors are highly heterogeneous. They present in terms of intra and inter-tumor heterogeneity at the genetic, epigenetic level. This has been well described by the most recent next-generation sequencing efforts, specifically at the single-cell level. And lastly, these tumors have a very limited response to traditional and targeted therapy, which is what I'm going to focus on today relative to the PTEN tumor suppressor gene. Now, my lab is really, as Clark mentioned, we focused on two genes which really account for a majority of GBM. The epidermal growth factor receptor, which is mutated in about 60%, or amplified and mutated 60% of all GBM cases, and the PTEN tumor suppressor gene, which is mutated about 40–45% of all GBM cases. Now, the reason that I'm very interested in these two genes is that they're intimately linked. So if you look here, you'll see the EGF receptor and this other version of it, EGFRv3, which is a highly oncogenic version of the wild-type receptor, they engage several downstream signaling pathways, notably the RAS-MAP kinase pathway, as well as the PI3 kinase AKT pathway. Now, sitting at this nexus between PI3 kinase and AKT is the PTEN tumor suppressor gene, which, as I mentioned, is predominantly recognized as a lipid phosphatase. So PTEN takes the product of PI3 kinase activation, which is PIP3, converts it back to PIP2, and thereby blocks the activation of AKT. Now, you probably know that AKT is an essential serine threonine kinase, which has a number of downstream effectors which lead to cell survival, growth, and proliferation. So you can see this is a very important component of the tumor cell. Now, PTEN also has other functions not really well understood in the cytosol. There have been some publications that show that it has protein phosphatase activity towards these substrates shown here, but as I mentioned, this is well less understood. Now, here's the problem. Now, we know that EGF receptor is highly involved in GBM. About 10 to 20% of patients who have been treated with EGF receptor inhibitors actually have a response to these inhibitors, despite the fact that EGF receptor is highly present in these tumors. And so we and others have tried to figure out why don't GBM patients do well on EGF receptor blockade therapy. And there are many reasons for this. One reason is the early generation EGF receptor inhibitors don't traverse the blood-brain barrier very well. The other reason, or one of the other major reasons, is illustrated here by Ingo Mellenhoff back in 2005, where he showed that genetically, those patients who were PTEN wild type, as I showed you on the previous slide, that blocks the AKT pathway, PTEN wild type, those patients had the longest time to progression, about 10 months on average, being treated with an EGF receptor inhibitor. And those patients where PTEN was deleted had the shortest or no response to therapy, as illustrated here. So lacking PTEN, you don't get blockade well enough of this pathway to respond to an upstream inhibitor. Okay. If you look closely at the data, there was a problem. There was about 10% of the cases that, despite having wild-type PTEN, did not respond to therapy. And so this caught our eye, and we wanted to figure out what was going on here. Why don't some of the patients, even though they had the hallmark of response, why don't they respond to therapy? And so we reasoned that perhaps PTEN's function was being commandeered by some other mechanism that was mimicking deletion. So I've cartooned here a number of post-translational modifications on PTEN that we and others have identified that both positively and negatively regulate PTEN's function. But there's been only one modification that I'm aware of that's been shown to regulate PTEN's function in a tumor setting and negate its activity, and that is phosphorylation by the SRC family kinases. Now, this was shown quite nicely by several groups at MD Anderson, but it was all done in breast cancer. And what they showed was that in breast cancers which were HER2 new positive, this led to activation of SRC family of kinases. I'm not sure which SRC member they were looking at. This would then result in PTEN phosphorylation on tyrosine. And when PTEN was phosphorylated, it would relocalize away from the cell membrane and away from its lipid substrate. So PIP3 is the lipid substrate for PTEN, and here in the cytosol, it was not able to dephosphorylate PIP3. And this led to a deregulation of proliferation, survival, and protein synthesis. And what they showed was that when you treated breast cancer with a HER2 new inhibitor, trastuzumab, otherwise known as Herceptin, this would then block the activation of SRC. PTEN was relocalized to the membrane and blocked the AKT pathway. Now, what they found was that in patients who were treated with Herceptin in conjunction with erythropoietin to ameliorate their anemia, it turned out erythropoietin would activate SRC and decrease the efficacy of Herceptin for those patients. So that was a problem. And then they could rescue those patients by treating with a SRC inhibitor, otherwise known as dasatinib. So this illustrated that SRC was playing a major role in attenuating the sensitivity to an upstream inhibitor, which was Herceptin. And so we reasoned that, well, maybe that's also occurring in those patients in GBM where PTEN was wild type. So we went forward and asked the question, where is PTEN phosphorylated on tyrosine relative to Src activation? And so this is work that was done initially by now a former postdoc in my lab, Tim Fenton, who's a faculty member at the University of Kent. And we published this a few years ago now. And what Tim did was to look in cells which had constitutively active Src, and we also did this by in vitro kinase assays, for where PTEN was phosphorylated on tyrosine. And we mapped by mass spec these 6 tyrosine sites as illustrated here. We're going to focus just on one of those sites today for several reasons. Number one, tyrosine 240, shown here, is highly conserved throughout evolution. Number two, we were able to make some really good reagents to this phosphorylation site. So we have at this time, it was a polyclonal antibody that specifically recognized it, as well as monoclonals that we now have. And thirdly, this site, in addition to being phosphorylated by Src, is also phosphorylated by FGF receptor, and very specifically by that tyrosine kinase. So FGF receptor does not phosphorylate these other sites. It's very targeted. And so we're just going to focus on tyrosine 240 today. And so with this information in hand, we wanted to determine if tyrosine 240, if it had a role in clinical behavior, if it does predict relapse, and is it an indicator of prognosis for GBM. And so the first thing we looked at was GBM clinical samples. So this was in collaboration with Paul Michel at UCLA at the time. And we looked at those patients that they had in their clinical trial at UCLA, and we saw a very nice concordance between PTEN expression. So 27 out of 33 patients who had PTEN expression also had phosphorylation of PTEN on that tyrosine 240. And this also correlated quite nicely with the presence of active Src in those same samples. And I think you could see very nicely in patient number 2. This is the region of the tumor microarray of that particular patient where Src was actually phosphorylated or active. PTEN is clearly expressed in that region, and phosphorylation of PTEN also occurs in that region. So I'm not going to go through all this data because it's been published for a while, but I'll just tell you the really important points that came out of this story. The phosphorylation of PTEN was associated with upfront resistance, and you can see here, these are the same clinical samples from that New England Journal paper by Ingo Mellenhoff, where we had responders versus non-responders, and these non-responders are PTEN wild type. You can see here that there's higher levels of phosphorylation of PTEN and active Src, and it was statistically significant. And the phosphorylation of this PTEN was indeed mediated by Src family kinases and FGF receptor in experimental models. This phosphorylation was also associated with acquired resistance. In the few cases where we could get pre and post treatment, so these are patients who are wild type for PTEN, we were able to see that there was an accumulation or enrichment of PTEN phosphorylation on that residue, as illustrated here. Albeit the numbers of patients we were able to compare was not very high, so we don't have statistical significance, there did seem to be a trend for this enrichment of phosphorylation on relapse. Next, we wanted to test if this was mechanistically linked to EGF receptor tyrosine kinase inhibitor sensitivity. And so what we did was we mutated this tyrosine to phenylalanine so it could not be phosphorylated, and we engineered it into mouse astrocytes, which were PTEN null, also expressed EGFRv3. And so we have wild-type PTEN and Y240F expressing astrocytes. We treat it with tyrosine kinase inhibitor targeting EGFR. And you can see here, looking at this relative cell viability assay, with increasing concentrations of erlotinib, which is an EGF receptor inhibitor, the cells which have the non-phosphorylatable form of PTEN, the Y240F, are more sensitive to the inhibitor. So blocking the ability to phosphorylate that site sensitizes the cells to the EGF receptor inhibitor. So it attenuates TKI sensitivity when it's wild type for phosphorylation. But this is where we really were kind of confused by the results. Even though PTEN was phosphorylated on this residue, it did not block its ability to act as a lipid phosphatase. So these are astrocytes in which we express constitutively active SARC, along with wild-type PTEN or the Y240F version of PTEN. And you can see that both versions clearly suppress the activation of AKT. So this is very different than the results they saw in the breast cancer scenario, where phosphorylation of PTEN led to elevated AKT levels. So this was very confusing to us at the time, and this is a point at which, you stop and you think. This is for the students in the audience, and for me, that we tend to get very bogged down to looking at where the light is brightest. We're very focused on doing one thing and getting very familiar with that one thing, which in my case was PTEN is a lipid phosphatase. It acts in the cytosol. It doesn't do anything else. But I don't think that's true. And what led us to think that it does other things is this result from the paper, that looking at patients who were wild type for PTEN and staining them for phospho-240, we saw that there was a clear difference in survival. Those patients where PTEN was phosphorylated had a median survival of 6 months, and those patients where PTEN was not phosphorylated, there was a median survival of 12 months. This is all based on PTEN phosphorylation data. The important thing here is what discriminates, what's the defining factor here is that all of these patients were treated with standard of care therapy, ionizing radiation, temozolomide, and the purpose of standard of care therapy is to generate DNA damage, which would kill the tumor cells. So this got us thinking that perhaps what we're seeing here has some role in repair of DNA damage. What got us really firmly committed to investigating this is the following data. So it has been shown that PTEN does have roles within the cytosol, within the nucleus, as illustrated here. Many labs have been studying this. However, there was one publication that caught our eye, and this is by Vuk Stambolik's lab at the University of Toronto, and he showed that PTEN deleted cells were particularly sensitive to ionizing radiation, so double-strand breaks. And so when he reconstituted cells with PTEN, this process was accelerated. And so we reasoned that perhaps what we're seeing here is this phosphorylation event on PTEN was acting as a switch which was regulating this repair process in some fashion. And so the questions I'll address and answer, I'll answer for you today are the following. Is the phospho-240 PTEN linked to radiation resistance in glioma? And how does Y240 phosphorylation regulate PTEN function? Can we target this mechanism to improve therapeutic efficacy, which is really the bottom line for what we do in my lab. And so the work I'm going to show you is by and large done by one postdoc in my lab, Jean-Louis Ma, one of the most talented postdocs I've had the pleasure of working with over the years. And so the first question that Jean-Louis did or answered was, do we see an inverse correlation of PTEN phosphorylation and the presence of double-strand breaks in clinical samples? And you can see here, we indeed do see an inverse correlation. So the more double-strand breaks we have, the lower levels of phosphorylation of PTEN on that particular residue. So it does seem that perhaps PTEN has a role in maintaining the integrity of the genome when it's phosphorylated on that residue. We also looked in a panel of clinical samples. These are GSCs, or glioma stem cell lines, from my colleagues at MD Anderson, Eric Suleman and John DeGroot. And we looked for the presence of PTEN expression, as shown here. And those that had low levels of PTEN expression tended to be radiation sensitive. Those that had high levels of PTEN expression were resistant, but only when they had FGF receptor expression. So FGF receptor and PTEN expression seem to go hand in hand in resistance to therapy. Now, I mentioned to you that PTEN does have function within the nucleus, so we wanted to look in those resistant lines, those GSC lines, which were PTEN positive and FGF receptor 2 positive. So here are 3 of those. These 3 were FGF receptor 2 positive, PTEN positive. We do indeed detect PTEN within the nucleus when we fractionate these cells. But in the sensitive lines, we see very little, if any, PTEN within the nucleus. Looking very closely at clinical samples at high magnification using our phosphospecific antibody, the entire amount or entire pool of phospho-PTEN is clearly within the nucleus. And here in the left column is an antibody that detects total PTEN that you see is throughout the cell. We could recapitulate this with either FGF receptor or SARC expressing cells, as shown here. Wild-type PTEN is clearly within the nucleus, and these cells expressing SARC, as shown here. With a total antibody for PTEN, you can see it's throughout the cell. So SARC or FGF receptor seems to correlate with nuclear phosphorylation and localization of PTEN. Now, to illustrate that this is mechanistically related to ionizing radiation resistance, we took U87 cells, which are PTEN null, expressed FGF receptor along with wild-type PTEN or the non-phosphorylatable form of PTEN, Y240F, and then treated these with ionizing radiation and did this clonogenic assay as shown here. And what you can see is that when we express FGF receptor, PTEN is indeed phosphorylated on that residue, and we see the greatest amount of surviving colonies in this assay. When we leave out, when we mutate that site to phenylalanine, we see this great reduction in colony number. And if we leave out the FGF receptor, we also see a reduction in colony number. We looked at different parameters of DNA repair. This is a neutral comment assay that allows us to look at the single cell level of the rate of DNA repair. You can see here the wild-type expressing cells have shorter comet moments after 24 hours of 10 gray radiation compared to the Y240F cells, which the comet tails are quite longer. So this illustrates quite nicely that the kinetics of repair are faster for the wild-type expressing cells. Looking specifically at the DNA repair process, was it homologous recombination or non-homologous end joining, 2 of the major DNA double-strand break repair processes in the cells? We use what's called a traffic light assay, which allows us to simultaneously get a readout for homologous recombination or non-homologous end joining based on GFP and M-Cherry reconstitution within the cells. And what we found was that the non-homologous end joining was unaffected by either wild-type PTEN or Y240F, but homologous recombination as monitored by GFP signal was enhanced by wild-type PTEN. So clearly, it's compromised when we don't allow PTEN to be phosphorylated with that point mutation. So HR seems to be the predominant process by which wild-type PTEN is affecting repair of double-strand breaks. We looked at the kinetics of repair as illustrated here, and what we found was that looking at in situ foci formation for double-strand breaks as monitored by gamma H2AX, we saw that the induction of double-strand breaks was equivalent in cells expressing wild-type PTEN as well as Y240F. Wild-type cells rebound back to baseline by 24 hours, so there is complete repair of those double-strand breaks. But the Y240F cells, we saw that there were persistent double-strand breaks out to 24 hours. Looking at RAD51, you could see that we have inefficient loading of RAD51, so we had far fewer RAD51 foci in cells expressing Y240F. So blocking PTEN phosphorylation led to a decrease in either Rad51 loading or Rad51 filament elongation. The process there, we're not quite sure of which one is actually occurring, but suffice it to say there were far fewer Rad51 positive foci. When we look at the activation of DNA repair, we did see that phospho-ATM is equivalent, both the wild type and Y240F cells, but there is persistent phospho-ATM as illustrated here in the Y240F cells, indicating that there's persistent double-strand breaks, and that is reflected by the presence of gamma H2AX by this Western analysis out to 24 hours, which is completely restored or eliminated in the wild-type cells. So what I've showed you so far, at least genetically, if we block phosphorylation of PTEN by that Y240F point mutation, we have persistent DNA damage, inefficient repair, we have non-productive Rad51 loading, and other parameters that we've looked at all seem to be normal, such as RPA, phospho-CHEK2, and phospho-ATR. So given that PTEN was localized to the nucleus when it's phosphorylated specifically on this tyrosine, we decided to ask the question, does it indeed bind to chromatin? And so you can see here, these are cells expressing wild-type PTEN treated with ionizing radiation, and we looked at the bound fraction of PTEN, this is on chromatin, over time. And you can see that PTEN is clearly bound to chromatin. It seems to be somewhat stimulated with radiation, but if you block the phosphorylation, there is no binding of chromatin by PTEN as illustrated there. Not only that, but the binding of PTEN seems to precede the loading of Rad51 shown here, which we do not see with Y240F. So PTEN, when it's phosphorylated, seems to have a chromatin binding ability. Not only that, but it seems that when it binds to chromatin, it's either promoting or it's selecting for more relaxed chromatin. So we looked at a marker of heterochromatin states. This is H3K9 trimethylation, which is a marker for heterochromatin. And we see that PTEN, when it does bind, the chromatin is much more relaxed. So there's less H3K9 trimethylation mark, which we do not see a change in for the Y240F expressing cells. So I think this might indicate that when PTEN binds to chromatin, it either promotes or is associated with a relaxation of the chromatin, which is presumably, and there's been several reports of this, is a better substrate for homologous recombination. Another way to look at the relaxation of chromatin is by micrococcal nuclease assay. So this is just treating chromatin with MNase and looking at the laddering of nucleosomes as shown here. And the Y240F cells have far fewer nucleosome liberation than we see with the wild-type cells. So there's a fair amount more of laddering of chromosomes, nucleosomes, indicating once again that the chromatin is much more relaxed when PTEN is bound to it. So next, we wanted to see if blocking the phosphorylation would indeed also eliminate PTEN's ability to bind to chromatin. So this is using PD1774, which is an FGF receptor inhibitor. So in the vehicle control, you can see here once again, after radiation, PTEN binds to chromatin, but using an FGF receptor inhibitor, we attenuate this binding, which mirrors the persistence of double-strand breaks as monitored by gamma H2AX, which we do not see in the absence of this FGF receptor inhibitor. We then turn to glioma neurosphere lines, which better recapitulate the biology of GBM. So in this particular line, HK281 is PTEN null, FGF receptor 2 positive. So this allowed us to reconstitute those cells with either wild-type PTEN or Y240F. And we did a limiting dilution assay to see if there was any effect just by this mutation in the ability of these cells to form colonies as in this assay. And you can see in the non-irradiated state, there's absolutely no difference in the wild type versus the Y240F expressing HK281. However, after radiation, you can see here that the Y240F cells had a much reduced ability to form colonies by this extreme limiting dilution assay, which mirrored the presence of double-strand breaks over time after radiation, which we do not see in the wild-type cells. We engrafted these cells into orthotopically into nude mice, and in the non-irradiated cells, you can see that the wild-type and Y240F expressing HK281 had the same survival kinetics. However, after a single dose of ionizing radiation, this is 5 gray, you can see that the cells expressing Y240F, those mice had an extended survival compared to the wild-type PTEN expressing cells. So Y240F cells are more sensitive to ionizing radiation. Looking at glioma neurospheres which have endogenous PTEN and endogenous FGF receptor, you can see that PTEN is indeed phosphorylated on tyrosine 240. We could block that pathway by either leaving out FGF ligand or blocking the pathway by using the FGF receptor inhibitor as shown here, and either one of those blocks phosphorylation of PTEN. We saw no difference in the ability of these cells to form colonies without radiation. However, if we radiate, you can see very clearly that blocking the phosphorylation of PTEN led to sensitization to radiation. They were very poor at forming colonies. And once again, this mirrors what we see with the presence of double-strand breaks as monitored by gamma H2AX. So blocking the pathway, we see these the double-strand breaks are not sufficiently repaired out to 24 hours in comparison to stimulating with FGF as shown there. Now, by this point, you're probably wondering, Frank, does this have anything to do with the enzymatic activity of PTEN, right? So PTEN is a well-known lipid phosphatase activity, as I started my talk. Does this activity of PTEN, is it needed for this ability to facilitate homologous recombination? And to do this, I turned back to some very old data that I actually did as a postdoc. So as a postdoc, I first started working with PTEN, looking at its enzymatic activity relative to mutations which were isolated from clinical samples. So these are all point mutations. I think there might be one deletion here that were isolated from GBM as well as cervical carcinoma. And I analyzed them back in this paper to see if they had lipid phosphatase activity. So this is a water-soluble lipid substrate. And you can see here, here's the wild-type PTEN, has very good lipid substrate, lipid phosphatase activity, but all of these PTEN point mutations were defective for that. So that, that gave me a number of mutant alleles to test for this ability to repair homologous recombination. So we picked one of them, G129R, and you can see here that G129R is still very capable of being phosphorylated on that tyrosine 240. And this is monitored with our monoclonal antibody. And here's the Y240F control shown in the middle. Now, does it still bind to chromatin? Indeed, it does. So here's the wild-type PTEN. Here's G129R. After radiation, you can see that the mutant, the lipid phosphatase dead form of PTEN is still very capable of binding the chromatin. And this is the control in the middle, the Y240F. And this binding to chromatin mirrors what we see for wild-type PTEN. We see the repair of double-strand breaks is facilitated by G129R, where the Y240F, we see this persistence of double-strand break after ionizing radiation. We looked at foci formation. This is by gamma H2AX and Rad51. And wild-type PTEN and G129R are indistinguishable in their kinetics of repair and their loading of Rad51. And then in looking at a clonogenic assay, this is where things got a little bit interesting, and I think we can explain why, that G129R and wild-type cells had the same number of surviving colonies after radiation. However, if you look closely at the size of the colonies in the G129R, they were about 2 times bigger than the wild-type cells. And what I think this means is that G129R has no ability to block AKT activation. Therefore, you still have this proliferative function within the nucleus, but within the cytosol, but within the nucleus, G129R still has this ability to enhance homologous recombination. And just to prove that, so G129R, shown here, versus wild-type cells, does not have the ability to block the activation of AKT. Hence, this is a permissive pathway in those cells. So I think this shows very nicely the separation of function of PTEN. There's a canonical pathway within the cytosol, which regulates AKT activation, but within the nucleus, there's an enzymatic activity independent of function which facilitates homologous recombination. So the whole objective of my lab is to try to bring basic discovery to the clinic. And so we wanted to see if this could be translated in terms of using FGF receptor inhibitors to treat GBM patients and then also treat them with ionizing radiation. So could we block the phosphorylation of PTEN orthotopically in these animals? So this is this is TS528. This is a PTEN wild type, FGF receptor 2 wild type endogenous levels. We engrafted animals with this glioma neurosphere line, waited for tumors to grow orthotopically, and then did a single treatment of this blood-brain barrier penetrable FGF receptor inhibitor, AZD4547. And we did one treatment and then sacrificed the animals over this time course, took the tumors out, immunoprecipitated PTEN, and looked for the phosphorylation of tyrosine 240. And what you find very clearly is that at 4 hours, we see a decrease of phosphorylation of PTEN, which is quickly restored at 8 hours. So this gave us an indication by this pharmacodynamic analysis when precisely we should irradiate these animals to have the maximum benefit to ionizing radiation. So we set up the following preclinical model. Using TS528, we engrafted animals, waited 12 days. We had imageable tumors orthotopically, and then did 3 days of this treatment regimen. AZD4547 followed by ionizing radiation at either 2 hours or 4 hours after drug, and we did this on 3 iterative days. And what you see here, by fluorescence molecular tomography, in animals which were treated with AZD followed by radiation 4 hours after drug treatment, we see a decrease in tumor volume or tumor burden by measuring by fluorescence molecular tomography. And this translates into an increase in survival. So only at 4 hours after drug do we see this increase in survival. At 2 hours after drug, with or without radiation, with or without drug, there's no difference in survival. So we have this window of opportunity, if you will, where we block the phosphorylation of PTEN, we can affect greater sensitivity to ionizing radiation. Now you're probably saying, well, that 4 hours is a very small window. It's probably not tenable. It's not possible in a clinical setting. And so we've now started I'll get to that in a minute. One of the things that the reviewers asked us was how well this is tolerated by animals. So we basically took non-engrafted animals and treated with ionizing radiation and AZD and followed their weight over time. And you can see versus control, there's no loss in weight. We did gross pathology analysis on a number of organs. We see no effect whatsoever. So by these parameters, it seems to be well tolerated. In engrafted animals, so these are animals with TS528, we looked for the presence of cell death by tunnel analysis. And you can see we only have appreciable cell death within the tumor and not within the surrounding area of the tumor. So the effect seems to be very specific for the treated tumors. Now, as I was mentioning, 4 hours is a rather short window of time to expect to use that drug in the clinic. So AZD4547, as I showed you, at 4 hours we see a great reduction in phosphorylation. So we looked at a number of other FGF receptor inhibitors, and you can see that these two here have very little suppression. This one here at 8 hours, this Janssen compound at 24 hours might be usable, but this compound here, TAS-120 from Taiho Pharmaceuticals, is very effective. It's an irreversible FGF receptor inhibitor, blocks phosphorylation at 8 hours, and at 24 hours it's still completely gone. We haven't, we're going to repeat this experiment and go further, but we think that this might be a much more usable compound compared to AZD4547. So with my colleagues at MD Anderson, we are setting up a window trial with GBM patients to use TAS-120 on recurrent GBMs. Okay, so at this point, I told my postdoc, John Wee, I'd really like to know if this modification of PTEN has any role in the normal development of an animal. So we actually went ahead and made a knock-in mouse. So we made a single mutation, Y240 to phenylalanine. This is throughout the entire mouse, and these mice had absolutely no phenotype, none whatsoever that we could detect. We haven't aged them extensively, but at least during development and normal adulthood, blocking the phosphorylation of PTEN had no effect on these animals. And isolating meths from these mice, you can see that the activation of downstream signaling, stimulation after EGF or serum, is completely identical in either wild type versus the Y240F. So this mutation had no effect on the ability of PTEN to act, once again, as a lipid phosphatase. But this is where things I thought kind of got interesting. So we made astrocytes from these animals. Actually, we crossed them onto an INK4AR phenol background, and we engineered these astrocytes to express EGFRv3 to make them tumorigenic. So here are the wild type versus the Y240F astrocytes, and we engrafted these into non-skid animals and did just a single dose of, actually 3 doses of 2.5 gray radiation, so 3 fractions. And you can see very clearly that one amino acid change conveyed sensitivity to radiation. We saw this extension in survival. Now, this is one of my most favorite experiments ever done. A single amino acid change is affecting the survival of these animals with these modified astrocytes. We also looked in other tissues of these knock-in animals to see if there are other avenues of sensitivity to radiation. So we looked in the distal colon, which is known to express FGF receptor and is known to be sensitive to ionizing radiation. So looking at the distal colon after 1 day of 8 gray radiation, you can see here that the villi length is equally destroyed in both the wild type and the Y240F littermates. At 4 days, the wild type mice have restored the villi, as shown there, but not the Y240F cells. Mice. We looked at the crypts within the distal colon, and you can see here the proliferation, as monitored by Ki-67, is more or less the same in both the wild type and Y240F mice. After 4 days of radiation, there is restoration of proliferation within the wild type mice, but we do not see that within the Y240F mice. And just to illustrate that there is phosphorylation of PTEN within the crypt cells shown here, we stained with Y240, and you can see here that there's co-registration of phosphorylation with the proliferation in the colonic crypts. So at least in the distal colon, this seems to be another tissue which is very sensitive to radiation if we block the phosphorylation of PTEN. Now, PTEN is clearly not a DNA binding protein, and so we wanted to address how is PTEN being recruited to double-strand breaks. There must be some protein or proteins which is involved in this recruitment. So we did mass spec analysis on PTEN-bound proteins when it was bound to chromatin, and we found just one protein which we think is facilitating this binding to chromatin. And it actually turned out to be Ki-67. And this is the typical biochemical analysis. So precipitating PTEN, we bring down Ki-67. But if you block that phosphorylation, we do not bring down Ki-67. In the reciprocal experiment, precipitating Ki-67, shown here, we only bring down wild-type PTEN, but not G129. I'm sorry, not Y240F as shown in the middle. And G129R as another control clearly can be precipitated by PTEN with Ki-67. If we knock down Ki-67 as illustrated here, we decrease the amount of PTEN that is actually bound to chromatin, as shown there. Now, to try to figure out where on Ki-67 PTEN is actually binding, we looked at the structure of Ki-67, and there were several domains that were suggestive that could bind a phosphoprotein. Actually, there were two domains. There is this forkhead-associated domain at the N-terminus, which is known to bind to phosphopeptides, and that was followed by this protein phosphatase 1 binding domain, PP1BD, as shown there. So those were the two domains that we suspected might actually bind to Ki-67. And so we made truncation mutations of Ki-67 as illustrated here, and we found that these two truncations, number 4 and 5, which lack the FHA and PP1 binding domain, they were defective for binding to PTEN as illustrated there. We then focused on this region with several more constructs, and what we found was that if we destroy the PP1 binding domain, so we split it in half in construct number 7, that was defective for PTEN binding. But construct number 8, which is just the PP1 binding domain, was still efficient at binding to PTEN as shown here. So we think that the interaction is occurring specifically through this little region here of PP1 binding domain. We next knocked down Ki-67 with 2 different hairpins, and then restored with these two constructs, number 6, which has the intact PP1 binding domain, and number 7, which the PP1 binding domain is disrupted. And you can see here that the restoration of PTEN binding to chromatin is mediated by construct number 6, but not construct number 7. And then using a peptide, so we made 2 peptides that map precisely to this PP1 binding domain, as shown here, a wild-type peptide and one where we mutated several of the amino acids. The wild-type peptide could diminish the amount of PTEN binding to Ki-67 in a transfection assay, whereas the mutated peptide did not disrupt that interaction. Importantly, when we use these peptides, we also mediate sensitivity to radiation. So the wild-type peptide, which disrupts the interaction, we see enhanced double-strand breaks as monitored by gamma H2AX. The mutant peptide, which fails to disrupt that interaction, we do not see that enhancement of double-strand breaks. So there's a possibility that in addition to an FGF receptor inhibitor, that we might be able to use some sort of peptide approach or other small molecules that can disrupt this interaction between PTEN and Ki-67 to enhance sensitivity to ionizing radiation. Now, okay, so there's a couple minutes left. So I just want to leave you with the following thoughts in terms of trying to understand maybe better ways to treat patients based on PTEN, the PTEN status of the tumor. So, I mean, this is essentially what we want to achieve. We want to be able to treat tumors and elicit cell death. But I think there are 3 states of a tumor cell that we need to think about in terms of PTEN status. When PTEN is wild type, it has these 2 functions, one of which is in the cytosol where it blocks AKT activation, and one within the nucleus which is enzymatically independent but binds to Ki-67, can facilitate homologous recombination. There's a second scenario where PTEN is mutated, where this process in the cytosol of blocking AKT is defective, but still retains the ability to elicit homologous recombination. And then the third scenario is when PTEN is deleted, AKT is once again deregulated, but there is no function within the nucleus because PTEN is missing. So all three of these have, I think, different aspects that we need to contend with. In the first scenario, I think an FGF receptor inhibitor coupled with ionizing radiation, as I've showed you, will be effective to mediate enhanced tumor death. In the second scenario, blocking FGF receptor inhibitor, but I think also an AKT or a PI3 kinase pathway inhibitor would be in order in conjunction with standard of care therapy. And lastly, in the third situation, blocking AKT should be enough in conjunction with standard of care therapy to yield the same outcome. And this has actually been well described by Vuk Stembolik's lab, that PTEN deleted tumors are much more sensitive to ionizing radiation. So I just want to acknowledge my fantastic team at the Ludwig and collaborators. So Jean-Louis Ma is the postdoc I mentioned who did the bulk of the work I showed you today. Rachel Takara is my technician who does all of our mouse work, actually made our knock-in mouse. I have to acknowledge Richard Kalogner and Waylon Zoe and Claudia de Albuquerque, fantastic collaborators at the Ludwig Institute who are mass spec aficionados as well as mismatch repair and homologous recombination experts. Of course, Clark and Jay were extremely helpful in providing assay systems to study homologous recombination, including the traffic light assay. John DeGroot and Eric Suhlman at MD Anderson, who are my clinical collaborators who are initiating the window trial. Laura Oriana, I didn't show you her data, but she actually did molecular dynamic analysis looking at how PTEN interacts specifically with Ki-67, and now we believe we have a structural understanding of the interaction and the region of PTEN via which Ki-67 is interacting. And lastly, Suli Marie and Theos Galatro, our collaborators at the University of Sao Paulo, who were very helpful in looking at clinical samples for the inverse correlation with phospho-240 and the presence of double-strand breaks. And of course, lastly, Tim Fenton, who started this work when he was a postdoc in my lab and is now at the University of Kent. He was once again, another fantastic postdoc that I've had the pleasure of mentoring. And with that, I'd be happy to take any questions, and thank you for listening.
Questions or comments?
I'm wondering in GBMs how often FGFR is activated by what mechanism, and at the beginning you said this Y240 residue can also be phosphorylated by SARC.
SARC, right.
So, how often do you have SARC activation without FGFR activation? Would it be better or necessary to also inhibit SARC? So you really focused on inhibition of FGFR to enhance radiosensitivity.
Right. So SARC could be downstream of FGFR for sure. So there's a composite problem there. FGF receptor expression was in a majority of the GSCs that we've looked at. So better than 50% had appreciable levels of FGF receptor. And every time we saw that, there was indeed phosphorylation of PTEN. So there was a definite good link there. We still have to, we're actually doing a much bigger panel of GBM patient samples that we're analyzing for the presence of 240 and the presence of either FGF receptor activation as well as SARC activation. And to this date, it looks like FGF receptor, at least in GBM, is the major player.
Just one other question about you showed 3 scenarios for GBMs at the end.
Yep.
In the middle scenario, you have active FGFR and
Yep.
phosphorylation at 240. And I'm kind of missed, how much of the PTEN ends up in the nucleus. Is enough drawn into the nucleus that you end up with PI3 kinase activation as a consequence of just...
No, the bulk, and we have that in the paper, we did fractionation. The bulk of PTEN is in the cytosol.
Still, okay.
Still, a very small amount gets into the nucleus, but everything within the nucleus, stoichiometrically, we've analyzed is phosphorylated on 240. There's very little of PTEN in the cytosol that's phosphorylated on 240, despite there being much more there.
One question regarding clinical samples. Do you see any correlation between PTEN phosphorylation in 240 and Ki-67? Any positive correlation?
Well, Ki-67 has got so many roles. We haven't looked at that. I mean, it's a proliferation marker, so I don't suspect there'll be any linkage between levels of Ki-67 or anything like that.
Yeah,
I mean, it has a major role. I didn't go over the usual function for Ki-67. It acts as a molecular surfactant, a chromosome surfactant. So it binds to the peripheries of chromosomes and keeps them from coagulating by eliciting a very positive charge on the outside of chromatin. So I think that is the fundamental function of Ki-67. PTEN binding to Ki-67, I think, is just a mechanism to cause a change in the chromatin state or localize changes in chromatin state.
I wouldn't expect it to. The Ki-67 stoichiometrically is much more abundantly expressed, and only a very small fraction is actually interacting with PTEN.
Yeah.
And so it wouldn't be it wouldn't be a worthwhile experiment to do.
Yeah.
So one more question about other phosphorylation sites. You focus on one. Do you think the others, do they have any other function in the IR sensitivity?
Yeah, we are making those composite mutations. So what I didn't show as well, we've done these phosphogels, which looks at the shift of a phosphoprotein in a gel based on its phosphorylation state. And when we mutate the 240 to phenylalanine, it all collapses down to a single band of PTEN. But when PTEN is phosphorylated on that residue, we actually see there are 2 sites on the gel, which might indicate that the 240 phosphorylation is a trigger for an additional site of modification. And maybe it's one of those other 5 sites of phosphorylation. We're looking into that. But 240 seems to be a key site.
I have 2 questions. First one is how many percent of patient has a Y240 mutation?
Well, so Y240 mutation has not been seen in patients.
Okay.
Yeah, but there's mutations that surround that particular residue.
Got it.
Clark and I are actually looking at.
This is not the therapeutic target at this time, directly.
It is. Pharmacologically.
Yeah.
So blocking the phosphorylation is definitely doable pharmacologically.
Got it. And when
there are mutations that affect phosphorylation of 240.
Got it. And the second point is that how this actual blockage of the double strand break repair affects the genetic stability in the meaning of antigen production or those kind of stuff.
Antigen production? You mean like neoantigens? Yeah. So yeah, that's an excellent question. We're looking at that. We've actually done we have RNA-seq data that we haven't fully analyzed, but maybe Clark can better address this. I don't know about defects in homologous recombination. Will that lead to neoantigens as much as if you had a defect in non-homologous end joining? Don't know, but
a lot of these proteins don't make it out of the ER. I mean, when they're mutated. So neoantigen quantity and quality is not a one-to-one correlation with DNA repair defects, because the DNA repair defects tend to generate specific type of events And many of these events results in essentially non-functioning proteins, and to have antigens, you have to have protein that's then processed by MHC complex. So it's not a one-to-one correlation. People like to think it, but simple-minded.
I understand. So the question is, if we, I guess, block the phosphorylation of PTEN, can we promote enhanced mutations that would generate neoantigens? Excellent question. Like I said, I don't know if we will do that from an HR defect and make the tumors hot. Yeah, that would be superb, but I don't think HR will really lead to that.
The one thing I would add to David's question is, in terms of EGFR, one of the things we're looking at is that we know FGFR activity differ depending on subtypes.
Subtype, right.
Right. And so, for example, the mesenchymal subtype tend to have the highest level of FGFR. I think that's a molecular, potentially a molecular predictor for a sort of FGFR inhibition.
Yeah.
And so it's, and in almost everything, I would actually say the 50% number sounds about right, but in the mesenchymal, in every mesenchymal model we looked at, FGFR has been very, very hot.
Mm-hmm. Yeah. Now, I might add to that, one of the things that we're looking at in other cancer types, are there other kinases that will phosphorylate this site? So we have done a tyrosine kinase screen through Connexus, which is a company that will take your peptide and it will run it through a recombinant tyrosine kinase screen. And there are other tyrosine kinases which will phosphorylate that site, and they put them in rank order. Now, FGF receptor is really at the very top, but there are other ones. I'll give you an example, like BMX is another kinase which will phosphorylate that site specifically. We haven't tested any of them yet, but there might be other cancer types where this mechanism of facilitating HR is important outside of GBM.
Thanks very much. Let's give him another thank you.